Okay, thank you very much for EDT to invite me for this presentation. I'm going to present Rhizobia research in South Africa. The first time I received this request or invitation to present Rhizobia research in South Africa, I was like, oh, am I going to go back in time to 70 years ago? All the research conducted in South Africa on Rhizobia. So how am I going to do that in 30 minutes? Then I have to take my own discretion. And the delegates in this room, I think you have to bear with me if I don't mention a very important research that you have made. It might be by chance. I know that a lot of you have done several good research, but in general, then we have to summarize it briefly in such a manner that which ones should be mentioned. So, this will be the presentation outline that I'm going to present. As I said, 70 years of Rhizobia research is a lot. So, briefly, I'll try to touch on biological nitrogen fixation, just an illustrative introduction. Then the early Rhizobia studies, starting from the 90s, 1901 up to 1990 or 2000, study of the taxonomy and phylogeny of Rhizobia, expansion of the South African Rhizobium collection, because a lot of research has been done ever since the inception of Rhizobia research in this unit, and then I will have a concluding summary and recommendations. Jonathan has touched in detail about the process of biological nitrogen fixation, which I'm not going to go to the details, but this is an illustrative introductory aspect about how biological nitrogen fixation takes place, and it has been summarized into three aspects here. The first one is root hair, then we have infection of the root hair, the second one, and the third one is nodulation. So the entire process of biological nitrogen fixation starts with a molecular dialogue. I think Jonathan tried to make a dialogue with somebody there. So the same thing happens in the process of nodulation. So the molecular dialogue starts with a secretion of the flavonoids by the plants, and these flavonoids are immediately recognized by the bacterial node D gene, and this node D gene then transmits this message to the other genes, especially these are the node A, B, C, or the common nodulation genes, and then these ones, they encode for an enzyme which produces a very important structure called the node factors, and these node factors are they are lipochitino-oligosaccharides, and these are the ones which start the initiation of the root node, the nodule formation here. Then once the initiation of the root nodule formation is started, then the final process with a number of other like cell divisions in the primordium and all that, then the nodule is formed. So what the bacteria do is that they travel all the way through the root, via the root hair, starting from the root hair, then the infection thread all the way into the nodule, and they become the bacteroids. And this is where the actual process of all this biological nitrogen fixation takes place. So in the bacteroids, we have the NIF genes, the nitrogenase enzyme, and the atmospheric nitrogen is converted into the usable form of nitrogen. So this is just briefly how biological nitrogen fixation takes place. So now I'm going to take you back in time to the 90s, 1901, 1906, because this is when the first South African Rhizobium research started. Although it's not a complicated or aspect of research, but some field trials were conducted even that time. So the first report goes back to 1903 by NOBS, when they were doing some kind of field trials with in Elsenburg in Stellenbosch on a forage crop or a forage legume called Serradella. And they had a problem of establishment of this Serradella, and finally they concluded, or they attributed this because of the lack of knowledge about the actual root nodule forming bacteria. And this is actually partly true because there is no significant Rhizobia research conducted in South Africa until the 1950s. So the first research on Rhizobium was initiated at the University of Pretoria and followed by the University of Stellenbosch in the 1950s. But actually, the comprehensive research on nodular Rhizobium symbiosis was initiated at the Agricultural Research Council, Plant Protection Research Institute, the then PPRI, now we call it PHP. So this was done with the establishment of the biological nitrogen fixation in 1965. So during that time, then important research focused on particularly nodulation, nitrogen fixation, yield improvement, field trial. So just to talk briefly about the research that time, mainly it focused on indigenous legumes such as, for example, Trifolium africanum, and other important indigenous legumes have been researched. So the nodulation status of this indigenous legume actually was started by first making a very extensive survey about the indigenous legumes. About 1,500 indigenous legumes, close to 100 genera have been surveyed, and also their nodulation status was compiled. Trifolium species that time was a very good legume, especially to do research. So Trifolium africanum at the Agricultural Research Council, researchers like Ian Lowe and Stridom, they conducted several field trials on Trifolium research, and they found out that actually Rhizobium leguminosarum is the key species for many of the Trifolium species, and it can nodulate different strains of Rhizobium leguminosarum can nodulate different Trifolium cultivars, but there is strict host specificity among the Rhizobium leguminosarum strains that nodulate the European clover, meaning that they are not effective on African clover. The other important indigenous legume was Medicago. Research on Medicago sativum was conducted, and they identified that Cynorhizobium melluleti mainly occurred on most of the agricultural fields where this important indigenous legume was planted. Stafford and Stridom in 1970s and D. Lejude in 1994, they conducted important research, both glasshouse and field trial. So the Cynorhizobium melluleti strains associated with Medicago sativum, they are also well distributed in the soils, and they can nodulate Medicago polymorpha, which is actually an introduced lucerne. So the final conclusion they got from this research is that there is effective nodulation of uninoculated Medicago sativa and the annual Medicago truncata in this soil because of the establishment. So in many cases, there was no need to inoculate because of the establishment of the Cynorhizobium melluleti strains in many of the fields where lucerne is grown. Coming to other important legumes, cowpea and groundnut. These strains of Bradyrhizobium species, which are indigenous to South African soils, are widely distributed in many of the soils where these crops are. So it is very rare to see some kind of response to inoculation. So you do some, even with the most important effective inoculant strain XX21, we call it, the initial one was CB75, then you inoculate and you don't see any inoculation response. So this is because of the establishment of the Bradyrhizobium japonicum, which are specifically nodulating this legume. Stridom, in 1987, they conducted widespread field trials, and especially with peanuts, with highly effective strains under irrigation at 2 sites over successive seasons. They didn't see any kind of yield increase in this as a result of inoculation. So there is no difference between the uninoculated and the inoculated controls because of the establishment of the rhizobia. Coming to soybean, which is one of the most important researched legume, can say protein legume in South Africa. In 1976, Jensen, Rensberg, and colleagues, they did a survey and finally found out that South African soils lacked the Rhizobium strains required to nodulate soybean. So this triggers the need to evaluate exotic strains of Rhizobium for their performance, for the efficiency of nodulation and nitrogen fixation. And because of this very widespread selection, trials have been conducted by importing some exotic strains, and in the 1960s, Rhizobium strains were released. New Rhizobium strains were released for commercial use on soybean. The first released strain was the WB66, but this was to be discontinued in 1966, and followed by another strain, WB61. This one again discontinued in 1978 and replaced with WB1, and this was again has to be replaced by another one, because from time to time, after a certain period of time, these strains, they lose their efficiency and also their performance. So in the late 1990s, 20 strains of Rhizobia were evaluated for nitrogen fixation on 30 local soybean cultivars, particularly initiated by research scientists at the Agricultural Research Council. And this resulted in the release of Rhizobium japonicum strain CB1809, which we call WB74 as a soybean inoculant. As you can see, at that time, researchers led by Ian Law, the late Jacomina Bloom, and Staffis Stafford in 1990, they announced, they broke the news on this Farmers Weekly magazine that a new soybean inoculant has been released, which is WB74. Here on this picture, you see the late Jacomina Bloom was also responsible for one of this important research and the coming into being of this strain WB74. And Dr. Staffis Stafford, here you see him applying the WB74 inoculant into the furrows of the soybean field. And this, I can say, is one of the pioneer discoveries when it comes to inoculant formulations and product development in South Africa. Then, this strain has to be released into a field where Rhizobium japonicum strains have been established for the past almost 40 years. So for 40 years, we have different inoculant strains, but some of these inoculant strains might have established in the soil. And now we are introducing WB74. So what's going to happen is that there must be competition between WB74 and the already established strains 40 years ago. So then there needs to be a study to confirm the efficacy and the nodulation competitiveness and the effectiveness of this. A comparison was done, nodulation occupancy trial was done, and it has been found out that WB74 or CB18 was superior to 60 to 73% of the established isolates from different soybean farms, including Bergville, Morgensen, and Codescorp. Seed inoculation in furrow application of this strain results in nodule occupancy 3 times at Bergville and Morgon as compared to the other established strains. So this work has been published by research teams Wilhelm, Julian, Jafta, Bloom, and also Ian Law. It's a very good research output, and it should be published. So this is my presentation on the brief introduction about the early research on nitrogen fixation, yield, and maybe strain selection. There is also a very important research which has been done in the past, in many years, by the group of Felix Dakora at the moment, University of Pretoria. So since the early 90s, Felix Dakora has done a lot of research contributing, again, particularly to small-scale farmers. And compared to strain selection, his research mainly focused on nitrogenous activity, the leg hemoglobins, nitrogen fixation, estimation studies, and the like. I just have a couple of slides about this, and here you can see that the Thank you very much. So, in this slide, we see the effect of added nitrate on nitrogenous activity of Bambara groundnut and Kirsten bean. This research tells us that available soil nitrogen or nitrate has a detrimental effect on nitrogenous activity. And you can see here that days after adding of this amount of nitrate, the nitrogenous activity is decreasing. So it's a known fact that available soil nitrate is inhibitory, and we have to also know that when there is no nodulation, then, or when there is little nitrogen fixation, we have to look into the many different limiting factors. Jonathan has described a number of factors, but also the soil available nitrate is a very important thing to consider because it is detrimental. Another important research by Felix Dakora is to investigate if there is any relationship between nitrogenous activity and the leg hemoglobin concentration. So the higher leg hemoglobin concentration, the more the nitrogenous activity and nitrogen fixation. Another important science-led research output, which was conducted by many of, especially the students with Felix Dakora, is the symbiotic nitrogen contribution in various indigenous South African legumes using what we call the natural 15 abundance technique. So using this technique over the past many years, I'm just mentioning here a few, they have tried to estimate the amount of nitrogen which is contributed from nitrogen fixation. Because mind you, when we evaluate the nitrogen in the plant, it might come from both biological nitrogen fixation, also the available one. So we are not sure that 100% it is fixed nitrogen. So we have to calculate, and this is done by what we call the N15 natural abundance technique. So over the years, they have determined for Doralia in the Cape Fynbos, cowpea in town, Bambara groundnut in Mpumalanga, groundnut again, 3 legumes for acacia, albizia, and a number of others which I cannot mention at this very specific period of time. Now going into a different aspect of research, which is study of the taxonomy and phylogeny of Rhizobia. Because Tilvali has mentioned that the bacterial identity must be known. So in any case, whether we do formulation or inoculation, we have to know who is there, what is the active ingredient. And apart from knowing who is there again, what is the soil microbial diversity in terms of Rhizobium? So which group of Rhizobia are nodulating this? So in 1995, actually, before 1995, there were no such detailed research about the taxonomy of Rhizobia, but a group led by Dagutat, supervised by Prof. Stein, tried to investigate the taxonomy of Rhizobia, indigenous Rhizobia, using wholesale protein. Then in 2002, one of the most important research on this one, which we can say probably the first, was a polyphasic taxonomy of Rhizobia associated with legumes by Julian Jafta in 2002. It's a PhD study of Julian, again by Prof. Stein. What he did was he used 16S RNA sequencing, RFLP of 16S, intergenic spacer, NIF-H gene sequencing. The conclusion he got was that in many cases the 16S sequencing gives that the bacteria are mainly Bradyrhizobium and Mesorhizobium, but when he compared it with the other molecular technique, which is the RFLP 16S 20S, then you can see, well, this one for the 16S, but when is RFLP, you see other additional genotypes which are also responsible to nodulate these legumes of South Africa. Another one is Lebechia, and Lebechia is, it might probably not of very important botanical importance, but nowadays there is an interest even by Australian researchers and others to domesticate Lebechia by taking some of the seeds from here into Australia because of its nature. It's growing in a very infertile, sandy and acidic soil, so it can be a very important candidate for sustainable production of legumes or soil fertility. So the first of this research in South Africa, actually in 2008, was done as part of the master's program by my colleague Francina, and she has done a detailed aspect of the Lebechia phylogeny and taxonomy. I didn't include every finding here. What you see here is that Lebechia is promiscuously nodulated by a group of Rhizobia, Mesorhizobia, and Bradyrhizobia, as well as the Beta Rhizobia group, which are Burkholderia. So it is a promiscuously nodulating legume, and you can see here also different fingerprint patterns of the different samples collected throughout South Africa. Then we come to research on newly emerging root nodulating Paraburkholderia species. For the past many years, 100 and plus years, the root nodulating bacteria are considered as Rhizobia. So the Rhizobia, well, its taxonomy has changed from time to time. So the Rhizobia, initially we have one genus, but now almost 13, 14 genera of the Rhizobia complex, which you call the Alpha Rhizobia. So the reason why it's Alpha Rhizobia is they are all in the Alpha Proteobacteria group. But in 2001, researchers in France and the USA, they found out that a group of bacteria not in the traditional Rhizobia group were found to nodulate legumes. And this is Burkholderia here. And then later on, another bacteria which is not in the Beta Alpha Rhizobia group was Cupriviadis. So this group of microorganisms or bacteria are now termed as Paraburkholderia because, well, actually so the Burkholderia and the Cupriviadis, they are in the Beta Rhizobia, but not all Burkholderia are beneficial. Some of them may be opportunistic human pathogens. So to differentiate those bacteria from the beneficial ones, then a new genus has been proposed recently, which is Paraburkholderia. So here in South Africa now, a lot of research is being conducted by a team at the University of Pretoria, Steenkamp, Beckers, Fanus Venter, and others. They are doing a very important, nice research on Paraburkholderia, and I'm just trying to show only a couple of slides here, and this is the description of new Paraburkholderia strains, Paraburkholderia stridomina and Paraburkholderia steini. So what they did was they tried to study the taxonomy of 12 nodulating strains from the indigenous South African fine bush legumes, Hypocalyptus sophoroides. These are just very nice flowering legumes in the fynbos area. So what they found, the finding they found out, these are the isolates which have been tested, and as you can see here from this phylogenetic tree, then they have their own distinct clades, and even they are, okay, when they were tested using the average nucleotide identity and DNA hybridization, then they form a distinct of their own. And for this reason, a new species has been proposed, and the first groups have been proposed to be Paraborculdaria stridomina, species nova, just to honor Professor Stridom, and the other groups are termed as Paraborculdaria steini, again to honor Professor Stein for the work they conducted for several years here in South Africa, and the work has been published on this journal. Of course, we have also collaborated in this journal from our BNF team. Another important research on this was also done by another team member of the University of Pretoria, Lazarus, and they also described a new species called Paraborculdaria polydire, also from the fynbos. So in order to have a good understanding about this new emerging Paraborculdaria and beta Rhizobia, then you can have refer to this review papers. So the fourth one is a bit about the expansion of the South Africa Rhizobium collection as a genetic resource, because a number of research is also being conducted at the nitrogen fixation unit. And we have more than 3,000 strains of Rhizobia and Rhizobacteria in the collection, which are stored at -80 in ultra-freezer and lyophilized cultures. These cultures, they are, in many cases, are supplied to inoculant manufacturers, researchers, and the universities. According to the Registrars Act 1947, then we have to always maintain and preserve, and also we conduct quality control of products which are made of these strains. And apart from this, we also do a greenhouse screening, authentication, nodulation, authentication research. And as you can see here, Trifolium, Pisum, Medicago, Vigna, Lupins, and a number of these, from time to time, we try to do the authentication for nodulation of this strain, and we do identification of all of these strains which are important in agriculture. We also do the phylogenetic relationships of these strains, particularly those ones which are called the commercial Rhizobia strains, which are indicated in here. So I listed here almost about 15 of the most important strains which have been over the past many years here in South Africa as a commercial strains, and the WB74 is still there, and many of us are also wondering how WB74 is very important and still active since its release in 1998. So, what are the possible genetic attributes to the success of Bradyrhizobium japonicum WB74? Then several research have been done. Then we want to look into what are there, if there is some important attributes. So we conducted a whole genome sequence analysis of the WB74, and we found out that it has several important attributes or beneficial traits, and you can see all these nodulation genes are in WB74, and the nitrogen fixation genes here, and which are indicated here in the genome operon, the NIF-H, NIF-B, NIF-E, NIF-N. Not only the symbiotic gene, we also have other beneficial genes, which you can see here, the ACDS for abiotic stress, for phosphate solubilization, exopolysaccharide synthesis, for auxin biosynthesis, oxidative stress, and a number of others. So these are very important essential traits which are possessed by WB74. Other important research that we have conducted at the ARC is the Rhizobia that modulates the indigenous rooibos, Aspalathus linearis. As we know, rooibos is a very important indigenous legume, and it has been thought that it is nodulated by Bradyrhizobium strains or Bradyrhizobium japonicum for many, many years, but we have found a very different result on this. So starting from 2011-2015, we did a symbiotic property study in the glass house, and so here the glass house portrait of the lineages, and different bacteria are responsible to nodulate this, but we selected one bacteria which is Mesorhizobium strain RB16S, and we took it into the field, and in Clown William, so this bacteria has been included with a number of other strains, and we did a field trial experiment, and we also published the output of this research. As you can see here, rooibos or Aspalathus is nodulated by Mesorhizobium species, Rhizobium species, Bradyrhizobium here, and we have also the beta proteobacteria. So it is a very promiscuous nodulating strain, but Mesorhizobium are the most important ones. I'm just finishing. I have a couple of slides. Another important research that we are currently conducting is the symbiotic property of Rhizobium tropiceus ARCCs on PGMP. Actually, this is part of the work. Currently, a wide spread, an intense study on PGMP is being conducted as part of a PhD study by Francina, my colleague. And one important output that we found was that there is a strain which does not contain any node genes or any nodulation genes, but it forms these big nodules. And you can see here the control and the inoculated plant. And no genes, so we have seen the whole genome of this. So how does this happen? No genes, but nodulation. So in order to investigate this, we have now written a proposal to NRF in collaboration with France investigators to investigate the node factor independent nitrogen fixing symbiosis, whether this process of nodulation takes place independent of the nodulation genes. And finally, this is the last slide. We are currently doing a screening of Rhizobia for alleviation of drought stress and improved symbiosis, because with the global climate change, drought is becoming a very important factor which negatively affects nitrogen fixation process, nodulation, and all that. So we are busy screening strains in soybean and also in other legumes which are capable of nodulating and fixing nitrogen under drought stress. This is just a glasshouse layout from our experiment, and some of the results I just picked up here, and the ultimate effect is to screen potential effective strains that can be used in drought-stressed conditions. Just to conclude, for the past many years, Rhizobia inoculation technologies, strain selection, commercial inoculant development, taxonomy of indigenous Rhizobia, nitrogen fixation, and crop yield improvement in major food legumes have been the common research that we have been doing, or the previous researchers and scientists have been doing. So we need to do additional research which focus on bacterial traits or genes that improve the tolerance to environmental factors such as drought by both by the host legumes and their horizontal symbionts, which is very important. Many different research outputs could not go beyond the publication stage owing to the financial constraints or lack of sponsors for funding. So different research are done, but they are published and they are not taken further into application, and that in many cases is because of funding constraints. Product development and commercialization requires a lot of money, and we need sponsors, to help us then continue the research in such a manner that it can be used in the community for the good of the community and as well as the country. I'm very much sorry to take this because you have to understand that 70 years of research in 30 minutes is not fair. So, thank you very much.